Cancer is the second leading cause of death in the world, and in 2018 it accounted for 9.8 million deaths worldwide. We all know how prevalent cancer is today, but what is it? In simplest terms, cancer is the uncontrolled growth of cells. When cells gain the ability to break off from the tumor and grow somewhere else, things become deadly. This process, the epithelial to mesenchymal transition, is a key feature of metastasis, the ability of tumor cells to travel and populate at new sites. Epithelial and mesenchymal cells have major differences in both structure and function. Epithelial cells bind tightly to each other by various junctions. They have polarity, which prevents differentiation, and they are attached to a basement membrane, preventing motility. In contrast, mesenchymal cells can differentiate and move freely through the body. There are a variety of biochemical signaling pathways that initiate this transition. However, this presentation will focus on the WNT pathway. The WNT pathway activates genes that foster the change of epithelial cells to mesenchymal cells.
Thank you, Olivia, for that introduction. Now we're going to dive further into the WNT pathway. The WNT, or WNT pathway, is a pathway that controls cell polarity, cell proliferation, and cell fate. Now while I draw out the pathway, here is a short rap. Cancer ain't nothing to be trifled, but I got it in my sights, right in front of the rifle. Mic check, 1, 2, 3. Learning about that transition called EMT. It begins with that beta-catenin, building right up and killing E-cadherin. Cell loses contact, then it starts to flow, headed to that blood vessel wall to proliferate, then it starts to infiltrate, but there's a way to stop it with ease. Research in pathways like WNT. Alright, now back to work. So, this WNT pathway usually exists within two states, either off or on. First, we're going to discuss the off state. Within this state, there are two membrane proteins of interest. The first is LDL receptor-related protein, or LRP6, and, for naming reasons unknown, a protein which is a 7-pass transmembrane protein called frizzled. In the absence of WNT ligand, there is nothing bound to this receptor, and so we get the formation of this destruction complex. This destruction complex contains a lot of proteins, but two of them are of primary interest. The first is GSK3 and the second is CK1. These proteins are responsible for both phosphorylating and ubiquitinating beta-catenin. Upon doing so, unfortunately, the beta-catenin is destined for destruction in the cellular proteasome. When it's destroyed, it can no longer influence genes in the nucleus. So what ends up happening? We get these co-repressors that bind to TCF transcription factors and prevent the transcription of these genes. Some other proteins of interest make their debut here, namely axin, APC, and DVL, or disheveled. We're going to talk about this more in the on state. So in the on state, we finally get WNT binding to that frizzled receptor. When this happens, a signal is transduced across the membrane, and GSK3 and CK1 are recruited towards the membrane. Once they're recruited here, these kinases then work to phosphorylate the cytoplasmic tail of LRP6. Upon phosphorylation here and phosphorylation here, we get the recruitment of axin and disheveled. Once axin and disheveled localize towards the membrane, APC follows suit, and now this entire network of proteins which used to make up the destruction complex can no longer do so. That causes beta-catenin to proliferate within the cytoplasm. Once this happens, beta-catenin is then transduced into the nucleus. Once it does so, it displaces those co-repressors we talked about earlier and allows transcription of these target genes to occur.
Much of research surrounding the WNT pathway is how it can be deactivated in cancer cells. Because of this, research has moved towards targeting this pathway for deactivation in these certain cells. For example, current research includes such approaches as bio-based therapies that target this pathway by activating the apoptosis-inducing FADD or the FAS associated via death domain pathway. As seen here, certain viruses can be used to essentially initiate apoptosis, including HPV, hepatitis C, or HIV. In another example, clinical studies have shown that lithium chloride can be used to inhibit certain proteins in the cascade as well. These small targeting molecules can help to modulate this pathway. Other molecules work in a similar manner and effectively shut down this pathway. However, the difficulty in targeting the WNT pathway makes it difficult to develop such effective long-term strategies and therapies. Ultimately, more research is needed before any of these therapies can be used in human testing.